So I think one thing that we've heard with the excellent talks that started today off is that even once you get your IND going, there's going to be some challenges that you're going to have to face. So I'm going to touch on what some of these challenges may be and some strategies that you could employ in order to meet these challenges head on. I am a CMC reviewer in the Office of Tissue and Advanced Therapies at the FDA, and I'm focused on a gene therapy review, which covers a variety of products that all use a similar mechanism in order to provide therapeutic effects. Instead of focusing on specific challenges for any individual type of product, I'm going to talk about some broad challenges that we see across gene therapies in general. Although I will be using gene therapies as the example, because these are broad challenges, the principles I'm talking about are also generally applicable to cell therapy development as well. So last year, former FDA commissioner acknowledged what I think many people in the gene therapy field were already feeling, in that many of the challenging questions when it comes to gene therapy relate to product manufacturing and quality. At the FDA, we try to employ flexible regulatory approaches in order to allow for product development. Many of our sponsors take advantage of expedited development review programs, and that's what I'm going to focus in for the beginning of this talk, talking about the need for product characterization, and then the end of the talk is going to give more specific recommendations on comparability studies. So perhaps the gold standards in expedited development programs are breakthrough designation and then the newer regenerative medicine advanced therapy designations. So there are some differences in the prerequisites in order to getting these designations. However, what I'd like to point out is that the benefits of these programs are very similar between the two, where you can engage with the FDA frequently, including senior management, in order to make sure that your scientific and regulatory development programs are aligned so that you can have a smoother development process. As we just heard, though, these expedited programs do pose some considerable challenges on the CMC side, and this is particularly evident when clinical programs are advancing rapidly, and therefore the timelines from early development to late development may be compressed. And so therefore, we recommend that you employ some forethought and you start planning for commercial manufacturing early on, and this often does include comparability studies. So what kind of challenges are we talking about? And these are really across the board in all aspects of CMC development. Specifically, when your timeline is compressed, you may have limited manufacturing experience, including limited in-process testing and limited product characterization. And this is really exemplified in the rare disease space, where you may be able to conduct a complete clinical study using one lot of your product. And you could imagine that if you only have produced and characterized one lot of your product, that it's going to be difficult in order to define the process parameters and the critical quality attributes that are related to your biological and clinical effect. And so we really recommend that you explore these parameters during your clinical development process, that you do use more than one lot, and that you also pay attention to retaining enough samples so that you can retest and compare early and late stage material if necessary. There also may be issues with limited knowledge of impurities. You may not have a lot of stability information if the development process has been rapid, and this is really necessary for you to move towards licensure. And then kind of an undertone to all of these limits is that there's, in addition to limited product characterization, limited time for assay development. And so when you're coming in for licensure or for your later phase studies, you should be working with assays that are qualified, and you should also be developing reference standards concurrently with your product. As you may imagine, for many gene therapies, there's not commercially available reference standards. And so it's important for your product development to also be developing these reference standards. And so therefore, we are really encouraging you to do early product characterization so that you can understand the critical quality attributes of your product and the range that supports your clinical effect. And so what might this look like? Exploring multiple critical quality attributes early on. And we encourage you to include these on your lot release. You don't have to have specifications for them or release criterion for them. This is really a time for you to thoroughly characterize your product and get a good understanding of the attributes that relate to the clinical effect. And this will allow you to then take this wealth of information that you have collected early on in your development process to be able to choose the relevant lot release tests for late phase studies and commercialization. In addition to exploring multiple CQAs, we recommend that you explore multiple attributes or aspects of a specific attribute. And so this may be that you utilize a matrix of assays in order to get a better understanding of each attribute, or that you use orthogonal methods. And I'll have an example of this in a little bit. Furthermore, we recommend that you also include assays that are stability indicating so you can get good characterization throughout your manufacturing process and for shelf life determination. So I'm going to be talking about product characterization right now in the guise of why it's important for licensure. The second half of the talk, I'm going to then really bring this point home by why you need to characterize early in order to support comparability studies. So I had mentioned that oftentimes there are different assays that may be used to test a certain attribute. And so the example that I'm giving here is specific to AAV gene therapy vectors. It's a measure of purity where we recommend that you look at empty to full vector ratios. As you can imagine, the empty particles don't have DNA in them, so they are not going to be delivering your gene of interest. There are multiple assays that can be used in order to measure empty to full particles, and some of them may be more amenable to early stage trials or preclinical studies, whereas other assays may be more sensitive or specific or more amenable to commercial lot release testing. And so by exploring these different methods to understand what's best for your product and your process is encouraged throughout your product development. Perhaps the biggest challenge to our sponsors, though, is really developing a potency assay. Now, these are the regulations on potency, and I understand sometimes regulations have a lot of words. So I'm going to try and focus us in on what I think are really the important parts of potency that you should be exploring throughout your product development. So what is potency? Potency is the specific ability or capacity of the product as indicated by appropriate laboratory tests to affect a given result. And the tests for potency can be in vitro or in vivo that are specifically designed for each product. So very often, you can't take an assay off the shelf for a gene therapy product in order to determine potency. And so this takes a lot of time for our sponsors to really define and develop and refine their potency assay in a way that is useful for commercial lot release testing. So during a normal product development process, our sponsors generally would take a stepwise approach to both product characterization and assay development. And you can imagine that they would accumulate data over the different phases of the clinical trial. But what happens when you're in an expedited program, right? We just heard that this causes a lot of trouble when we're working on compressed timelines. So what's important to realize is that the regulatory requirements do not change when you're on an expedited development program. What happens is that you're losing some of that critical time that our sponsors often devote to assay development. And then this may cause a problem because you may have a gap in knowledge or either knowledge about your product or in technical expertise on how to define your product, which is going to really delay your ability to move forward to licensure. And so specifically for potency assays, when we're talking about gene therapies, there's a number of challenges that you may face while developing these assays. First and foremost, you might not know the mode of action for your product. So you can imagine that there's instances where the gene that's related to a disease is known. However, what the gene product does in the body is not fully understood. So without that knowledge, it's really difficult in order to develop a pertinent potency assay. There may be time constraints related to lot release testing, and this is particularly a problem for autologous products. There may be limited material available for testing. And as I touched on before, there may not be reference standards available, and so development of these reference standards would happen concurrently. And then I think we all have an understanding that cell-based assays are going to be a bit more variable and take more experience to control as compared to a more standard chemical assay. And so we really recommend concurrent and early assay development. So in this case, I'm going to use an example for CAR T cells. So for CAR T cells, we do recognize that the main mode of action is pretty well understood, right? Antigen-specific target cell killing is your mode of action. However, there's other attributes for your CAR T cells that contribute to this mode of action. These are multifunctional living drugs, as they're called. And so therefore, we recommend that you characterize a wide variety of product attributes as they relate to potency and the clinical effect and the biological activity throughout development. And so in addition to a cell killing assay, you may be looking at a cytokine profile from these cells. We know that CAR T cells engage in autocrine and paracrine signaling. There may be differences in T cell activation markers and cell expansion and differentiation in an antigen-specific manner is also important to understand how that relates to your product. And so what this will allow you to do, if you engage in early product characterization and assay development, is that when it comes time for commercialization, you'll have this wealth of knowledge in order to understand which assays are most beneficial for your lot release and commercial manufacturing process. So what does this look like in a normal development process timeline? So once again, I'm keeping it with phase 1, phase 2, phase 3. Understand that some of our sponsors may be skipping some of these phases or these timelines may be quite shortened. So we really recommend that you start characterization and assay development even as early as preclinical development if possible. And so first I'm going to tell you what happens if you don't heed this advice. So what's going to happen, and we've seen this with many sponsors, is that there's one assay that's used in preclinical development. However, that really does not translate well to the clinic. So then for the early phase studies, they may use a surrogate potency assay, such as gene expression. However, this is not going to be sufficient for late phase studies, in which case you then have to transition again to another potency test, which may not be quantitative or may not fully encompass the potency attributes of your product. And therefore, you may have to stall your product development process in order to develop this potency assay so that you can gain information and experience with it in order to inform your commercial application. What would be much more beneficial to your timeline for product development is to start these assays early, to gain as much information as you can early on and be able to leverage that information in the BLA. So as I said, the first part of my talk was really focusing in on how early product characterization is important as you transition towards commercialization and licensure. But what might come for many of you prior to a BLA is actually a comparability study, or more than one comparability study. And a comprehensive understanding of your product is going to be imperative for analytical comparability studies throughout your product development. So for the rest of the talk, first I'm going to talk about some general considerations for comparability and comparability studies, and then I'm going to go into a little bit more of specific examples of when comparability studies might be needed and what considerations are taken into the amount of comparability study that would be required. So first and foremost, what is a comparable product? A comparable product does not mean that the product is completely identical pre and post change. You made changes. It's very unlikely that they are going to be different in some attributes. The purpose of a comparability study is to show that the pre-change and the post-change product are highly similar and that the existing knowledge is sufficiently predictive to ensure no adverse impact on safety or efficacy of the drug product. So right in the middle of that definition is really hits home on why it's important to have early product characterization, because you have to have a certain amount of existing knowledge in order to understand your comparability study. So aside from us asking you to do comparability studies, why is it important from your point of view to do a comparability study? So the thing is that you have accumulated a certain amount of clinical data with your pre-change product, and you want to be able to use the data that you accumulated to support your post-change product. You don't want to just throw away all of that time and effort that you put in the clinic prior to that change. And so in order to be able to leverage that information, you need to demonstrate that the key attributes are comparable pre and post change. In order to start your comparability studies, you should analyze the attributes that are likely to be affected. The first part of this is a risk assessment. Once it's determined that a comparability study is necessary, that there is sufficient risk associated with the change, then you should start to develop a prospective protocol. Oftentimes these protocols include a side-by-side comparison of either retained samples or of split manufacturing, and this is really dependent on the product that you are developing at the time. Oftentimes if you're working with an autologous starting material, we do highly recommend that you use split manufacturing for your comparability studies. Part of this prospective protocol should be predefined criteria that includes a relevant statistical analysis that will set the criteria for whether or not you're going to determine if your product is comparable. And the statistical analysis should be based on lots that have been shown to be safe and effective if you have that data available. Additionally, we recommend that you're evaluating comparability at multiple stages of the manufacturing process, not just at lot release. For many of your products, you know that the product is very tightly related to the process itself. And so changes often in one part of the manufacturing process often affect downstream attributes as well. Dependent on your change, you may need to assess the impact on product stability. And dependent on the risk of the change or on the outcome of your comparability study, you may need to conduct additional non-clinical or clinical studies. And so due to this uncertainty of the information that you're going to need based on your comparability study, we highly recommend that major changes are made prior to your registrational study so that you're gaining the most impactful information with your commercial or proposed commercial manufacturing process. So we get a lot of questions on what do we mean by a relevant statistical analysis. So what you're trying to do is show that your product is comparable or equivalent. So we recommend that you use equivalence testing in order to analyze your comparability studies. I would like to say that oftentimes we get sponsors who suggest that a student t-test would be applicable for comparability testing, and this generally is not encouraged, as student t-tests are a test for superiority and not for equivalence. So that's getting a little bit into the weeds, but I just wanted to put it out there so you knew ahead of time. So I'm going to walk you a little bit through what you may experience with an equivalence test. The example I'm using is a toast. These tests are applicable to any comparability studies that you would be doing. So just to show how generally applicable it is, I'm going to relate it to comparing fruit, because I think that's a little bit easier to say. So what you're going to do is you're going to take a look at your product, and you are going to think about, using the data that you have on hand, what attributes are important for your clinical effect, and what range in that attribute supports the effect that you have seen. So going back to the fruit, all of these are fruit, right? But it would really take a lot for you to convince me that an apple and an orange are comparable. And I know that pineapple has apple in the name of it, but that is exactly where that comparability ends in my book. Okay? But for each attribute, you're going to develop a range of attributes that support your product comparability. So for some attributes, it might not matter whether or not you have a green apple or a red apple. And so you could see that you may have a larger range of variability that is acceptable for your product for that particular attribute. However, there may be attributes that have a very tight range that are related to your clinical effect. In that case, maybe only red apples are okay. And so you can imagine that your predefined variability limits for that CQA would be quite tighter. And this is a point where you may want to come in and talk to the FDA and get advice on your comparability study prior to performing the analysis. After you've performed the analysis, you then fit your data into these variability limits, and then you make decisions from there on whether or not the study concludes that your pre- and post-change product is equivalent, whether more studies need to be conducted, or whether they are not equivalent, and how that's going to affect your product development process. So when might a comparability study be needed? First and foremost, if you are going to make a manufacturing process change, particularly late in product development, we're going to recommend a comprehensive comparability study. And as I said, this is so that you can leverage the clinical data that you have generated pre and post change. This may be a change in reagent, a change in a process step, any number of things. So we do determine the requirement for a comparability study based on a number of attributes of your product, including whether or not it's in the early or late stage of development, if the change is determined to be a major or minor change, and then the perceived risk to the patients. And so that is something that you should determine with a risk assessment, and then you should come and talk to us about. I would like to acknowledge, though, that many times these changes are made because you want to improve your process, and we encourage you to do that. You should not feel like you are stuck with the process that you started your phase 1 study with. You may want to reduce a culture time or improve purity. What you do need to take into consideration with your comparability studies is that the attribute that you are trying to affect may not be the only attribute that's affected. And that is the point of your comparability study, to understand comprehensively how that change affects your product. We often see that the manufacturing facility changes as the product progresses through development. Oftentimes, this is a change from a more academic manufacturing facility to a commercial manufacturing facility. We really recommend that, if possible, you are able to identify and transfer your manufacturing to your proposed commercial manufacturing facility prior to those registrational studies. This change is often associated with changes in the manufacturing process itself, which complicates the switch in manufacturing facility and in your comparability study. And then, depending on your product, comparison to historical experience may not be sufficient, and we may ask for some split manufacturing or a more comprehensive comparability study. So maybe you aren't changing manufacturing facilities, but you're adding on more manufacturing facilities. And in this case, the point for your comparability study is a little bit different. It's not to compare your pre- and post-change product, but it's to support the fact that you are manufacturing the same product at multiple sites. And therefore, the evaluation of the clinical data that you are generating can all be combined and analyzed together in order to support your licensure. And in this case, we recommend a comparability study regardless of the stage of development. To facilitate your comparability studies, we recommend that you identify a single reference site and that this site is used as the comparator for all additional sites. To reduce variability, you may institute the same SOPs, reagents, equipment, whatever you can to reduce variability between these sites. Additionally, even at early stages, we would recommend that you set some sort of defined lot release criteria even for your characterization studies. And this is in order to try and support comparable manufacturing and the same product being manufactured across your sites to help your clinical analysis later on. Okay, now I know I'm talking about comparability studies, and that has to do with your product. But since I have you here, I'm just going to talk about one other thing, and that's if you make changes to your analytical tests. So this does not require a normal comprehensive comparability study as we've been talking about. It requires a bridging study. And so an example of this, the one that I think is most prevalent right now, is probably related to dose determination for viral vectors. AAV vectors in particular is what's coming to mind. And so if you're changing your assay, and this may be that you're changing from a more non-specific primer probe set in a qPCR assay to a product-specific primer probe set, or perhaps you're changing from, for instance, a qPCR assay to a ddPCR assay. You should conduct a bridging study in order to make sure that you are providing your patients with a consistent dose. So you can imagine that if you change your assay mid-clinical trial, and then all of a sudden you realize as you're analyzing your data later that there was a difference in the dose that was being administered pre and post assay change, that this could really complicate your analysis later on. Similarly, we recommend that you conduct bridging studies if you have multiple testing sites that are conducting the same assay, or if you're changing facilities for your testing. Oftentimes this is accomplished by using side-by-side testing of the same material. Once again, it's important to have retention samples in order to support these bridging assays. And then depending on whether or not the assay is part of your stability plan, it may impact your stability studies. So these are just things that you should think about as you're moving your product and process development forward. Okay. In summary, gene therapy products are complex biologics, and multiple assays may be necessary to be developed in order to fully characterize the critical quality attributes that are associated with gene therapy products. Therefore, we recommend that you engage in early product characterization and assay development, and that this can be a continual process throughout the product life cycle. And this will allow you to have a thorough understanding of your product's mode of action and how the CQAs relate to the clinical effect. Oftentimes, gene therapy product development includes changes that require comparability studies. And we encourage you to come talk to us to understand when comparability studies are necessary and to what extent they should be covered. But our recommendations are often related to the type of change that you're proposing, the stage of development, the impact on CQAs, and the impact on interpreting clinical study data. So I hope that this provided some useful information for your product development. Just as a shameless plug, we are hiring people, so if you would like to come be my coworker, come talk to me. This is my contact information. I'd like to thank you for your time and also for everybody who helped contribute to this talk.
Thank you very much for that presentation. We probably have time for one question.
Hi, so in the cell and gene therapy space, you face fast timelines. You maybe go phase 1, 2, and then you enter pivotal because it's a small rare disease population. You also have a lack of animal models for cell-based therapies. So given these two kind of dichotomies, if you're switching manufacturers from the academic center to, say, a commercial CDMO, and that timing coincides with your pivotal trial start, how do you navigate CMC comparability along with your clinical development timelines?
So our recommendation, the question has to do with aligning your CMC development with your clinical development. And so we really recommend that once you start to get some positive clinical data, that you start to think about your commercial manufacturing process then. And so if possible, you could then move to your proposed commercial manufacturing facility prior to starting that pivotal study. If that's not possible, then come talk to us. It's going to be a product-specific advice, but we would recommend The use of multiple engineering runs, the use of multiple lots in your pivotal trial, maybe not the whole pivotal trial can be conducted with that, but as much that could use your commercial process as possible would be helpful.
Thank you.
Yep.
So I think we are getting kicked out for the next session, but Kimberly, if you're around and others have questions, and
Yeah.
you can follow up with her directly. Thank you.
Thanks, everyone.